Hi everyone, it's Mrs. Milne and welcome to your lesson on microscopy. All right, so remember, as you go along with this presentation, you can stop or pause it at any time so that you can jot down the notes at your own pace. So this word microscopy, well, you've got a picture here, so I think you can guess what it's about. It's about using microscopes, and specifically the type of microscope we use in class is called a compound or a light microscope. So in biology, we can't really see everything we need to see that is living or details on living things. So we use microscopy as a way to increase the size of a specimen. So typically in a lab setting, you have a scientist who is observing either a whole specimen or cells of a specimen that they can't see with their naked eye. So they put it under a microscope and woo, look at that image. So here we have an electron scan of a fruit fly, something that is so minuscule, but under a microscope we can see very, very fine details of that specimen. So you can see all of the bristles on the thorax, on the head here. You can see parts of the eye, and look at there's like little bristle-like structures on the eye that you wouldn't even know were there if you didn't have this microscopy or use of microscope. So there's a couple of terms we need to talk about with microscopy. One of them is magnification, and as you can see to the right here, we've got a magnifying glass. And what does that do? Well, it makes images larger than they actually are. So here we have some different magnifications of an object. So with the naked eye, we see this group of tree or population of trees really far away. And then we see 4 times, 6 times, 8 times, 10 times, and 20 times. So this would probably be with binoculars to increase the size of an image when you're standing very far away. With microscopy, we're able to look at things we cannot see with the naked eye. Here with the trees, we can see them. But in microscopy and specifically biology, we need to look at the structure and function of cells. Another term we need to talk about with microscopy is resolution. And as you can tell from A and B, A is very in focus and B is very blurry. And this measures the resolution, so how clear something is under the microscope. Sometimes your phone cameras will take a very clear image, so you say that it has a very good resolution. And sometimes when you're taking those selfies, they're a little blurry, so you would say that it has a bad resolution. Now I have a question for you. If you have a microscope that's really good at magnifying things, but is really bad at resolution, could you use it? No, because what you're looking at would just be very dark and blurry, and so therefore you couldn't see the details that you want to look at. Or let's say you have a really crystal clear image, but it's too small of an image. Could you use that? No. So you need a larger image to identify the structures on your specimen. So it's really important to have good magnification and good resolution with microscopy. So I keep on talking about magnification. Well, your microscope has different magnification, and we like to say, what's the power, or what power are you looking at under the microscope? So let's talk about a few lenses that are on your microscope. So first of all, you have something called an eyepiece, and the eyepiece is right up here. And that eyepiece always usually has a 10 times magnification. Then you've got 3 more lenses on your microscope that are called objective magnifications, and they're located right here. And you have a 4x, a 10x, and a 40x. And what that x means is times. So how many times larger is the image than with the naked eye? So you're looking through 2 lenses with your microscope. So in order to figure out the total magnification, you need to do some calculations. So total magnification is taking the eyepiece times the objective lens. So we're going to do a little bit of basic math here. So if we take a look at the next slide, we've got calculating magnification, and here's our total magnification. So our eyepiece is always going to remain 10 throughout the whole entire calculation. But our scanning objective, or our objective, will change. And this first objective, the low power, is 4 times. And this is the number we're looking for right here, that whole number. Not these decimals down here, but this whole number, this 4. So to figure out total magnification, we take 4 times 10, which is 40. Our middle power, or middle objective, gives us 10 times the magnification. So we take 10 times 10 equals 100. And then our highest power objective lens is 40. So we take 40 times 10, which is 400. And that's how you'll figure out the total magnification of your microscope. So now let's go through more pieces of the microscope. We've talked about the eyepiece here. We've talked about the objective lenses down here. So there's other important parts you need to know about. One of them is the arm. The arm is located right here, and the arm is what we use to grab to hold onto the microscope if we need to carry it around. We don't only grab the arm, but we also grab the bottom of it, which we call the base. And on the base, you probably have an on-off switch that might be located right here or might be located on the side. So you need to make sure that you turn that on so it turns the illuminator on. Another word for illuminator would be light. So the light then shows through something called the diaphragm. So right here is our diaphragm, and you can adjust the diaphragm, which adjusts the amount of light shown on your slide that you're looking at. Okay? The slide goes on something called the stage, and this is a special newer microscope that we have in our classrooms. It has a mechanical stage. So you're going to have some knobs on there that will move the microscope stage or move your slide left, right, up, or down. And for those of you that don't have this mechanical stage, you'll just have to do that by hand. You'll have to move your slide up and down, side to side, to look at an image under your scope. All right, so once you put something in, you need to adjust it, adjust those lenses so that it's in focus and that you have clear resolution. The two things you're going to use for that is your coarse focus, okay, coarse focus right here, which is the bigger knob, or your fine focus, which is the smaller knob down here. The coarse moves great distances to the stage, and we only use this one on the lowest power. And then the fine focus is used on all 3 of those powers or objective lenses. I kind of like to think about it as a cheese grater. You've got coarse grating and fine grating. Coarse gives you nice thick pieces of cheese, whereas fine gives you very tiny pieces of cheese. So coarse will move that image very high or low, and fine will move it just small distances. All right, so using this light microscope. So when you first use the microscope, make sure to be on the lowest objective lens, the smallest one there. As you can see, I'm pointing to it right there. And then you want to make sure that you move the slide over so that the specimen of what you're looking at is directly in the field of that light there. So let's watch this video here. Okay. You have to move that mechanical edge there so that it gets a nice good grip on that slide. And here you can see I'm moving over the slide so that it's right in the direction of the light. All right, so that's on the lowest power. Here's another view of it. Okay, so we're going to look at some colored threads, and this is the first thing you're going to look at. So let me go back, hit play here. So you have to open up that little anchor there, and then you can move the stage around using the mechanical knobs. This is the coarse adjustment knob to move it up so that it is in good view through the eyepiece. All right, so when you're changing, watch this video here, when you're changing objective lenses, you hear a little click and it tells you it's in place. And notice I'm on the medium power and I'm only using the fine adjustment knob. And then again, as we move to the high power, don't use that coarse adjustment knob, only that fine adjustment knob to bring it into focus and to get a good resolution. Okay? Well, I hope you've enjoyed your lesson on microscopy. Have fun in the lab and start exploring the microscopic world of life.